Welcome students to exercise 31, which is just one of the exercises that focuses on really cardiovascular phys. So we did the heart anatomy, and now we're going to dive into kind of the physiology of the heart. And one of the first things we're going to look at is what we call ECGs, electrocardiography, and the intrinsic conduction system, which is really kind of looking at kind of the electrical activity of the heart. So you guys are going to get to learn how to read an ECG. But before we get to the kind of new stuff, I just want to kind of briefly review how muscle works. So you have to remember, start with our resting membrane potential, right? It's about negative 70 millivolts. We get sodium influxing, potassium effluxing going out, and that kind of sets this resting value. And then before we can have an actual contraction of the muscle, a shortening of the muscle, whether it's a skeletal muscle, cardiac muscle, smooth muscle, doesn't matter, we have to have a change in that membrane potential. And so we do so by kind of opening and closing the ion channels for sodium and potassium. And so before contraction can occur, the membrane potential has to depolarize. So it has to get closer to zero or even above zero by opening particularly sodium channels, allowing sodium to influx. In cardiac muscle cells, it's actually both a sodium channel and a calcium channel, and so you get kind of a longer depolarization period, this kind of weird plateau phase, which makes tetanus impossible. In skeletal muscle, tetanus can occur where the muscles contract and then kind of get locked in that contracted or shortened position. If your heart did that, your heart would contract, but then stay in that contracted position, and it would not be working as a pump anymore. And so we're looking at both sodium and calcium to kind of make this plateau area. But we do have to have this depolarization occurring before we can get contraction. And when we look at ECGs, that is what we're looking at, is the depolarization, the electrical signals in the heart. The intrinsic conduction system is this series of what we call pacemaker cells. These have what we call autorhythmicity. They can self-depolarize. They don't need innervation from the nervous system to say, hey, it's time to depolarize. Skeletal muscle needs that, right? You have to get a signal from your brain out to the skeletal muscle to say, hey, skeletal muscle, change your membrane potential, so that leads to contraction. Well, the heart doesn't need that. You can pull the heart away from the autonomic nervous system, and it will still beat because of this intrinsic conduction system, because of these pacemaker cells. They're non-contractile, so they themselves don't contract, but then all the rest of the heart muscle contracts. But because we have this intrinsic conduction system, we get a very kind of stepwise depolarization of the cardiac muscle, the myocardium. This is one of the things that allows your heart to pump as a functional unit. It's not the right atrium contracts and then the left ventricle. It all happens kind of in a simultaneous, kind of coordinated fashion. The atria contract, well, the atria depolarize, and then the atria contract. The ventricles depolarize, and then the ventricles contract. And having this kind of series of nodes, series of or sequence pattern of these pacemaker cells really ensures that we have this very coordinated system. There are 5 parts to the intrinsic conduction system, or at least 5 major parts to the intrinsic conduction system. Starts with the SA node. From the SA node, the signal travels out and then to the AV bundle, atrioventricular bundle, where it pauses for a little bit, the AV node, then into the AV bundle, then to the bundle branches, and then up the walls of the ventricles in the subendocardial network, or Purkinje fibers. Let's look at the figure, because I think that's helpful. So here is the sinoatrial node, or the pacemaker. This is what kind of starts this whole depolarization process. It sets the rate of depolarization, so it sets the rate of eventual heart contraction, what we would consider a heartbeat. From the sinoatrial node, the signals spread through the atrial septum into the left atrium, but then also down the right atrium to the AV node, or the atrioventricular node. The signal will pause here at the AV node for a very brief period of time, which will allow the atria on both sides to kind of finish contracting to get all of the blood out of the atria and then into the ventricles. From the AV node, the signal travels into the AV bundle, which is where it finally comes into the ventricles here. And once it's in the interventricular septum here between the two ventricles, it splits into the bundle branches and then goes up the Purkinje fibers or the subendocardial network, and that's really what ultimately leads to the depolarization and then thus the contraction of the ventricles. So when we're looking at an electrocardiogram, an ECG or an EKG, what we are looking at is kind of a composite of all of the action potentials generated by the intrinsic conduction system, by the different parts of the intrinsic conduction system, and all of the contractile cells of the heart at kind of any given time. We're not looking at the contraction of the muscle, we're only looking at the electrical current. And we're also not looking at just a single action potential, we're really looking at kind of the whole big picture. There are kind of three major waveforms that we see on an ECG. The first is the P wave. What's occurring at the P wave is the depolarization of the sinoatrial node, and so it's really the depolarization of the atria. In the QRS complex, it's the depolarization of the bundle branches and the Purkinje fibers, and so what we're looking at is ventricle depolarization. This is also the period where atrial repolarization will occur, where it goes kind of back to its resting value. And then the T wave, the last one, is then the ventricles repolarizing kind of back to their resting value. So here's our P wave. Again, this is depolarization of the SA node, and so it's depolarization of our ventricles. The QRS is the depolarization of the ventricles. This is also where atrial repolarization will occur. We do not have a separate wave for it because the repolarization of the ventricles, because the repolarization of the atria, because the ventricles are so much bigger, their depolarization kind of hides or shadows the repolarization of the atria, and so you don't really see it. All you really see are the big ventricles depolarizing, so it kind of hides the repolarization of the atria, but it would occur under this QRS complex. And then our last wave is ventricular repolarization, the ventricles repolarizing, and that's known as the T wave. We can look at several different kind of segments or intervals. We have the PR interval, which is really looking at from the time the atria start to depolarize to the time they finish repolarizing. The QT interval, which is from when the ventricles start to depolarize to the time they're done repolarizing. We have the QRS complex, which is the ventricles pretty much depolarizing altogether. And then we have this flat segment here called the ST segment. And it's flat because, again, ECGs are looking at the electrical activity, not the mechanical contraction. In this segment here, this ST segment, this is where or when the ventricles actually contract, between depolarization and repolarization, right? Contraction follows depolarization. But because we're looking at a mechanical event, contraction, it doesn't appear as a wave on the ECG because it's not an electrical event, it's a mechanical event. So let's kind of follow the depolarization and repolarization through the heart as it kind of compares to the waveforms on the ECG. So here's our SA node. Remember, it spreads across to the left atrium, and that's our P wave. So as the signal travels from the SA node to the AV node and over to the left atria. right? That's that initial P wave. Once atrial depolarization is complete, again, the signal kind of pauses here at the AV node. That allows the depolarization of the atria to cause then the complete contraction of the atria to get all of the blood out of the atria and into the ventricles. The signal then starts to spread down the bundle branches and up the Purkinje fibers. That's our QRS complex. You'll see as the ventricles start to depolarize, the ventricles are repolarizing, but we don't see it under the QRS wave because the QRS wave, the ventricle depolarization, is such a larger electrical event. But by the time we get to the S, right, it's completely depolarized the Purkinje fibers, and the ventricles are now starting to repolarize. That gives us our T wave, and then at the end of the T wave, the ventricles are repolarized, and we're ready to kind of start the whole cycle all over again. When you look and when you read an ECG, you look at it initially and it just looks really complicated. There's all these different kind of segments along the ECG, and it can get really confusing. But what you have to realize is that typically when you do an EKG or an ECG, the standard number of leads, the little nodes that you actually place on the body, get placed on the different parts of the body. You can see all of the different ones that they go here, right? So on the shoulders, on the abdomen, and then across the various parts of the chest. All of these leads are looking at a different part of the heart. So imagine the heart is in the middle of the room and you have CCTV cameras from all around the room looking at the heart that's in the middle of the room. Well, that's what these different leads are, is they are looking at the electrical activity of the heart from all the different parts of the heart. And that's why, say for instance, if we were looking at this particular ECG, we're actually looking at 3 heartbeats, not, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12. We're not really looking at 12 heartbeats. We're looking at 3 heartbeats from various angles. So let's look at it again. So just like this example here. So this example is also looking at 3 heartbeats from various parts of the heart. You'll see that the way the ECG is broken up on the paper, they're labeled for whatever lead was placed on the patient's body, and then we have these 3 over here that have Roman numerals. Well, these leads over here are the actual leads that are placed on the patient's body. These ones with the Roman numerals are kind of a composite or an average of the various other leads. So sometimes these Roman numeral leads are referred to as virtual leads because they're really more of a composite, an average of the information garnered from the various angles of the heart. You'll notice that at some points the waveform is up, and the same wave, this wave here is the same wave as this wave here. The waveform is heading down because the parts, the leads are looking at kind of different parts of the heart. The depolarization signal may be heading towards the lead or away from the lead, towards the part of the heart that the lead is looking at versus kind of traveling away from the part of the heart the lead is looking at. And that's what then determines whether the wave is up or down. So if you have an upward deflection, that is depolarization heading towards the part of the heart that the lead is looking at. If you have a downward deflection, that is the depolarization kind of heading away from the part of the heart that is looking at. And that's why the same waveform, or the same for one heartbeat, might be an upward deflection on one lead, but a downward deflection on the other lead, because that at that same period of time, the signal is heading towards one lead, but heading away from another lead. And that's why we get those kinds of changes. One of the things that we can determine when we look at an ECG is we can look at heartbeat. Or heart rate. We look at the timing between the two heartbeats by looking at, and let me go back here for a second since this one doesn't have any of the paper, but if you look very specifically at this paper, it has a very specific kind of grid pattern, and because of that grid pattern, we can get the distance between two heartbeats, and then use that distance between two heartbeats to then calculate a heart rate. And that's one of the things that you guys are going to be doing in lab this week. The other thing that we can kind of determine is by looking at the kind of patterns in the ECG, whether the waveforms look normal, whether the intervals are longer than normal or shorter than normal. We can at least kind of intuit whether there's been damage to the heart or whether there's been damage to that intrinsic conduction system. So some of the things that we can look at is the different intervals. So if this PR interval is longer than it typically should be, it could mean that we have what we call a heart block, where the signal kind of gets stuck at the AV node, and it takes longer for the atria to repolarize or depolarize, and we get the signal kind of gets stuck there, and it doesn't travel very well then into the ventricles. So that's why it's called heart block. If that happens, the ventricles do have their own kind of self-depolarization, but they don't depolarize at nearly a good enough rate to kind of sustain life. So heart block can be quite dangerous. If the QRS interval is the kind of longer than it should be, what this means is that typically your ventricles are enlarged. They're bigger than they should be. Maybe we've got some congestive heart failure or something where the ventricles have kind of gotten a little too big. The reason we can kind of intuit that the ventricles might be too big is that if the QRS complex interval is longer, it's taking longer for the signal to travel through the ventricles, and so either it's getting kind of stuck along the way, or the ventricles themselves have just gotten bigger. If we look at the ST segment, again, remember it's flat because this is where the contraction of the heart muscle occurs. After depolarization, before repolarization, but if that segment is either too long or too short, what we might have is ischemia. Ischemia means that the heart itself is not getting enough oxygen, and then there's a problem with the heart muscle itself, not necessarily the intrinsic conduction system, but with the muscle itself. And then finally, if we look at the QT interval, from the start of the Q to the end of the T, that means if that interval is longer, it means that it's taking longer for, typically, that it's taking longer for the ventricles to repolarize, to kind of go back to their normal resting state, and that can mean that you might have, or typically tends to mean you have a greater risk of arrhythmia. Any kind of arrhythmia, A means without, so without rhythm. So anytime you have these kind of things like heart blocks or ischemia or anything like that, longer QT intervals, well, sometimes they're called junctional, there's things called junctional flutters, there's all kinds of weird ways that ECGs kind of get screwed up, and that's an arrhythmia. Whenever the heart is not pumping in a coordinated fashion like it's supposed to, in a rhythmic fashion, then that's an arrhythmia. So typically this means that if we have problems in the intrinsic conduction system, then it means that both the contraction and the ventricles of both the atria and the ventricles are not going to kind of occur in like a very coordinated fashion. And if arrhythmias kind of get too bad, we can get into a situation called Infibrillation, which is this really, really rapid, really kind of screwy, irregular contraction type of process. And it can occur both with the atria or the ventricles. If it occurs in the atria, it means the atria aren't contracting as a unit. But since the atria is mostly for getting receiving the blood and kind of getting it into the ventricles, that's not nearly as bad as, say, ventricular fibrillation, because if ventricular fibrillation occurs, then since the ventricles are what is pumping the blood out to the rest of the body, if they're not pumping in a coordinated fashion, then they're really not pumping the blood in any sort of efficient manner, and then if they're not getting the blood out to the rest of the body, then the rest of your body isn't getting oxygen, including your brain. And that's when they'll take the little AEDs and they'll use the little electrical nodes to kind of shock your heart back into a normal sinus rhythm. So here is a nice normal sinus rhythm, typical roughly 70 beats per minute. This one here is what we call a junctional rhythm. This is where the SA node is non-functional. The SA node is not functioning properly, and so you'll notice there are no P waves. At that point, the AV node can take over, but the AV node pumps at a slightly, or depolarizes at a slightly lower kind of rate, 40 to 60 as opposed to roughly 70, but still functional. This is the heart block. This is when the AV node the signal kind of gets stuck there. So we get a couple more P waves because the SA node keeps trying to send these signals to the AV node, and the AV node's kind of not really conducting them into the ventricles. You see the heart rate has slowed down quite a bit because at this point the ventricles will take over, but the ventricles pump and depolarize at an even lower rate than 40 to 60, and we do have a bit of a problem there. This is ventricular fibrillation, and you can see there's no rhythm to it at all. The ECG waves are completely and utterly chaos, and so at this point they're going to take the AED and they're going to give you an electrical shock to try to get your heart back into some sort of at least semi-normal rhythm if they can't get you all the way back to a normal sinus rhythm. Two other things that we can look at with the ECG is imbalances in the heart rate itself. So tachycardia is when you have an abnormally fast heart rate, if it's more than 100 beats per minute. And if tachycardia gets really bad, then you can go into a fibrillation situation where we're back to the total chaos and the heart really need to be shocked. And then the other one is if it's too slow, which is less than 60 beats per minute, and that's bradycardia. Bradycardia is a problem because if your heart is pumping too slowly, then it's probably not getting enough blood out to the rest of your body, and then we have not enough blood and oxygen getting out to those body tissues. However, bradycardia can actually result from endurance training. If you do lots of endurance training, you get your heart pumping really well, you work out your heart becomes very, very efficient, and it can actually, because it can pump more strongly, because the muscle itself is stronger, the rate of the heart can slow down and still get adequate blood out to the rest of the body. And so sometimes bradycardia it's like a desired result, but if you have if you're not one of those people who does lots of cardio training and you have bradycardia, you probably have a problem. So a normal heart rate is anything between 60 and 100, anything over 100, tachycardia, anything less than 60, bradycardia. And those are all of the different things that you guys are going to get a chance to look at whilst you are looking at ECGs this week in lab.